Hello, 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 hello. Hello, 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 and welcome मेरे सभी बहुत ही बहुत प्यारे प्यारे से and all the amazing students of PW English Medium. So am I properly audible and visible to everyone? So just message me once if I am properly audible and visible to you all. So today we are going to start with the class 11th first chapter, that is Cell, the Unit of Life. In the last week, if you remember, we discussed the chapter Sexual Reproduction in Flowering Plants, which was the first chapter of class 12th. Hello Kavi, how are you? Am I properly audible and visible? So just give me a thumbs up once if I am audible to you all. All the viewers, would you like to just give me a thumbs up that if I'm properly audible and visible to each and every one of you? Today is going to be an amazing session where we are going to cover the chapter Cell, Unit of Life, first chapter of class 11th, and very, very important, very, very simple chapter. Purely we are going to do NCERT based, your 12 CBSE pattern based, where I am going to make you revise all the simple, easy, complex, all the topics of class 11th Cell, the Unit of Life, with the help of your NCERT screenshots. So whole soul, my purpose of today's lecture is to make sure that within a limited time span, you people are able to revise this chapter from your NCERT. All the points I am going to cover today in this lecture. And yes, this lecture is going to be very, 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 very helpful to you all. So just make sure that if you start from the beginning, you continue till the end. Right? So once again, without any delay, let's start with the chapter Cell, the Unit of Life. Purely from NCERT point of view, purely question based lecture this is going to be, and post lecture I'm going to upload a DPP form where you can just practice questions from where you can people practice questions as well. Chalo, okay. So without any delay, shall we start? Is everybody in? Yes? Okay, very good. So let's start with the chapter Cell, the Unit of Life. Today we are going to cover up all the major topics of this chapter. First, starting with cell. What do you mean when I say cell, beta? So cell is nothing but the basic unit of life, basic structural and functional unit of life. The term cell was first given by scientist Robert Hooke. So first is, you see, Robert Hooke. This is Robert Brown is something different. Okay, so first cell, the cell terminology, the term cell was first given by Robert Hooke, right? While he was working on cork cell, which he saw under simple microscope. So what he got to see under the microscope? He saw some honeycomb-like structures. He saw some honeycomb-like compartments to which he referred to as cella. And this cella means cell, right? Yes or no? Hello, hello, Sirthi. S. Tirthi Moti. It's such a big name, I am not even able to pronounce. I don't know, is it really an English term or not? So yes, hello, 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 hello. Hello to all my dear students. So first of all, let's start. We have just started the chapter Cell, the Unit of Life. So everyone just pay attention and keep listening to me. If you have your NCERT book, then please keep it open in front of you with a pen, so that whatever important point I say, you can highlight it in your NCERT as well. So this lecture is really going to help you a lot in revising the important concepts of this chapter. And yes, so let's continue. So I was telling you, beta, what is cell and who discovered it and who gave this term cell. So cell is nothing but the basic unit of life. We can say that all living organisms, without weightage of the chapter is like, you can imagine, we are starting class 11th with cell chapter. So you can imagine that you are starting class 11th with cell. Otherwise, it is living world chapter, right? If you see your NCERT book, then the first chapter of NCERT is living world, followed by biological questions, biological classification, followed by plant kingdom and so many other chapters. However, we start class 11th with cell chapter. Why is it so? ऐसा क्यों है? Because this chapter is very, very important. So we find this chapter very important from the NEET point of view, from your future point of view. That is why we start class 11th with cell. So when we start chapter, when you start class 11th, we start with this chapter cell itself. Tells you that this chapter holds a great weightage in your class 12th boards as well and in NEET as well. Right? Okay, fine. So let's continue. Basically, beta, cell is the basic unit of life. When I say this, I want to convey that all living organisms, without any exception, they are made up of cell. Or you can say that anything which is not made up of even one cell is not considered living. So next point what you can write is anything not even made up of one cell is considered living, is not considered living. That means for any object to become living should at least made up of one cell. Ma'am, how many questions asked? Tell me, ma'am. Beta, at least 2 to 3 questions every year are supposed to be asked from this chapter. However, the number can even increase, but bare minimum is at least 2 to 3 questions. So please pay attention. This chapter is very, very important. And if you focus and if you study properly, then definitely it is going to help you a lot. So anything not even made up of one cell is not considered living. So basically, if we say cell is the basic unit of life, I want to convey that any object which is at least made up of one cell is living. The term cell was first given by Robert Hooke while he was working on cork cell. So when he cut a thin slice of a cork and when he viewed that thin slice under the microscope, he saw some honeycomb-like structures. He saw some honeycomb-like compartment, right? To which he called it as cella, and from here the term cell came. However, beta, Robert Hooke observed the first dead cell. Therefore, the credit to the discovery for the first living cell goes to Antony von Leeuwenhoek. He saw and described the first living cell, right? Now, like my body has a brain, your body has a brain. Similarly, a cell has also a brain which controls all the cellular activity. So the brain of the cell is known as nucleus, which was first discovered by Robert Brown. So you have two Roberts over here. Don't get confused. One is Robert Hooke, another is Robert Brown. Robert Hooke is credited for coining the term cell while working on cork, whereas Robert Brown is responsible for discovering nucleus inside the cell, which is also called as brain of the cell. Why? Because it is nucleus which controls all the cellular activity. So what is written in your NCERT? That Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Now you see over here, beta, when Robert Hooke was working, when Antony von Leeuwenhoek was working, at that time, microscope levels were not that great. They did not have high magnifying power lenses. So they could only observe the external structure of the cell. But with time, with more discoveries, with more invention came electron microscope, ultra electron microscope, which gave the power to the scientist to look deep inside the cell as well. And with the time, you know, with the discovery of electron microscope, ultra electron microscope, scientists got the opportunity to study the structural details of the cell, like cell membrane, cell wall, inside the nucleus, all the cell organelles, cytoplasm and everything. Correct? Now, let's continue to cell theory. So, let's talk about what is cell theory. So, before all these ultra electron microscope and before we go inside the cell, let's learn what is cell theory. So basically, beta, there were two scientists by the name Theodor Schwann and Matthew Schleiden. These two scientists, beta, Matthew Schleiden was the botanist, Theodor Schwann was the zoologist. And these two people in two different countries in two different years one working on plant cell, another working on animal cell. They both came to the same conclusion that be it plant body or be it animal body, all are made up of cells or the product of cell. So Matthew Schleiden and Theodor Schwann gets the credit. Ma'am, NASA is using your... Acha, wow, 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 wow. Thank you, Falco. I mean, if you are here just to praise me. So yeah, thank you so much, Falco. Yes, sir. And if you are interested in studies also, then I think I should continue. Matthew Schleiden and Theodor Schwann, these two people, beta, individually, independently, one working on plant, another working on animal. They found some common postulates that all living organisms are made up of cell or the product of cell. That is, cell is the basic unit of life. And this together formulates the cell theory. Apart from this, beta, Theodor Schwann also stated one very important information that animal cells, they only have one outermost boundary by the name plasma membrane. But when it comes to plants, then plants, apart from plasma membrane, they have one more additional layer outside the plasma membrane by the name cell wall. So it was Theodor Schwann who mentioned that plant cells have cell wall, whereas this cell wall is absent in animal cells. So let's read it out. In 1838, Matthew Schleiden examined a large number of plants and observed that all plants are made up of cells and their product. So what is the product of the cell? Tissues, organs, etc. At the same time, Theodor Schwann, a zoologist, he studied different animal cells because he was a zoologist. So he studied animals and reported that animal cell has the outermost layer, which is plasma membrane. However, he also stated in his studies that if I talk about plant cells and plant tissue, then apart from plasma membrane, as I told you, they also have the presence of cell wall, which is a unique characters of plant cell only. On the basis of which cell theory was postulated, these are the two important points of the cell theory that says all living organisms are made up of cells and the product of cell, and hence cell is the basic unit of life. Now, when you see this whole cell theory, you find that this cell theory is somewhere incomplete, right? Because you do not come to know that from where the cells are arising. So the modern cell theory says, or you can say this primitive cell theory was finally given the shape, or it was modified by another scientist called Rudolf Virchow. Rudolf Virchow was a Swedish scientist who added one big statement by the name Omnis cellula e cellulae. Now this statement states all living cells arise from the pre-existing cell, which means if I have one parent cell, then it is going to divide to produce new daughter cell. Further, they are going to divide and this process is going to continue, which means every cell has their own lineage, which means what? Every cell has their own lineage. None of the cell arises de novo. De novo का मतलब है कि खुद से. तो कोई भी cell खुद से नहीं बनता. Every cell has their own parent cell and they arise by the division. So this was stated by Rudolf Virchow. That gives the final shape to the cell theory. And now the cell theory adding the statement Omnis cellula e cellulae is also known as modern cell theory. So if the question comes, who proposed cell theory? So your answer is going to be Schleiden and Schwann. But if question comes, who formulated the modern cell theory or who gave the final shape to the cell theory? So your answer is going to be Rudolf Virchow. Why? Because he gave a very closing statement, that is Omnis cellula e cellulae. I hope it is clear to everyone. Cell theory, yes or no? Now, this is all about cell. Who discovered cell, living cell, dead cell, cell theory. You have studied so many points related to a cell. But now, let's discuss the overview of a cell. When I say overview of the cell, I mean to say, let's discuss the shape, size, structure and function of the cell. So there's a very common statement that your NCERT quotes, beta, cell can be of various shapes, sizes. It's not like if I have drawn a cell which is circular, so all the cells are going to be spherical in shape. No. Some cells can be long, elongated, some can be spherical, some can be like cuboidal, some can be like star shape, any shape. Cells can be of any shape and any size. There is no standard shape or size of a cell. This is a highly variable entity. So cells can be of various shapes and size. First point. Second point, always remember beta, shape of the cell decides the function it performs. Siddhi, pay attention. Second very important point, if you read your NCERT, it says shape of the cell decides the function it performs. Decides the function it performs. Like for example, you all know human RBC. हाँ सिद्धि बोलो बेटा. See, human RBC. What is the shape of the human RBC? It is spherical, biconcave disc-like. Why it is spherical? Why RBCs are not long, elongated? Why? Because RBC's function is to carry oxygen, right? In the form of hemoglobin. So if it is going to be long elongated, there are chances that RBC carry less oxygen. So therefore, because its function is to carry oxygen and it wants, and single RBC wants that at one time it should carry maximum oxygen, because of which its shape is spherical. Had it been triangular, had it been long cylindrical, it would have carried less oxygen. So to carry maximum oxygen, its shape has become biconcave disc-like. Similarly, tracheids, you see this diagram, this tracheids, why they are long elongated pipe-like? So basically, they are pipe-like only because their function is to conduct water and mineral. Imagine you have a tanki at your home and a motor. So how are the pipes laid down? Those PVP pipes, what is their shape? They are long elongated now. Why? Because their job is to conduct water, and water can very efficiently be conducted if the pipes are long, hollow from inside, elongated. So similar to PVP pipes are the function of the tracheids. Therefore, tracheids are also long elongated. So you can see over here, depending upon the function, it is the shape of the cell that is decided. Now, Siddhi is asking me a question, ma'am, what about viruses? Basically, remember beta, viruses do not follow in the category of cell theory because viruses are neither purely living nor purely non-living. We say that they are the connecting link between living and non-living. First thing. Why? Because viruses are acellular. Their body is not even made up of one cell. Hence, they cannot be considered living according to cell theory. So, when viruses are outside the host body, they are inert, inactive, dead. But as soon as they enter inside the host body, they take over the machinery of the host cell and then they replicate, multiply, increase their number, etc. So, viruses do not fall in the category of cell theory as it is not purely living, not purely non-living. I hope I am very much clear to everyone. Right? So what is the overview of the cell? Cell can be of various shapes, size. Hello ma'am, I used to watch your past recorded. Thank you Pavan, thank you so much for coming again on this platform. So if you are still preparing, you can just listen to this lecture of Cell, the Unit of Life. So here I am telling you an overview of the cell. Cell, as I told you, can be of various shapes and sizes. Depending upon the shape, function of the cell is decided. You can see over here different shapes. Red blood cells, as I told you, RBCs, they are biconcave disc-like. WBCs, they are irregular in shape. Then you have columnar epithelial cell. Your nerve cell, it's like star-shaped, the cyton which you say. Then you have tracheid, which is long elongated so that it can conduct water as efficient as possible. And some mesophyll cells of the leaf, they are round and oval. So you can see there are so many different vivid shapes and sizes of the cell. Now for the size, you can study your NCERT in one of the page. You see a diagram of different cells and their sizes. Virus sizes giving standard, bacteria size, then PPLO size, which is the smallest known cell, and the RBC is 7 micron in diameter. Longest cell is the nerve cell, largest is the ostrich egg cell. So all these data please learn from your NCERT, and even if that data is not correct or not updated, still if the question in NEET will be asked related to the size, they are only going to consider the answer which is printed on their NCERT textbook. So do not go too much into the absolute size of different cells. Just remember the data which is printed there in the NCERT textbook. That's it for you. Now, there are two types of cells, beta, basically depending upon the type of nucleus. If nucleus is primitive type, then such cells are called as prokaryotic cell. And if the nucleus is advanced type, then those type of cells are called as eukaryotic cell. So how many types of cells do we have? Two types, prokaryotic, eukaryotic. So here, first I am going to discuss the prokaryotic cell, their structural details, functional aspect, everything, and then we will switch to eukaryotic cell. Correct? So first heading is going to be prokaryotic cell. Pro means beta primitive. Pro means primitive. Karyo means nucleus. Okay? So cells which have primitive nucleus. Now, when I say yes, Siddhi, all bacteria are prokaryotes, absolutely true. Now, beta, when I say primitive nucleus, what do I actually mean? So I mean to say a primitive nucleus is such a nucleus which is naked in nature. Naked DNA means that the nucleus is represented by, naked DNA means it is represented by only one circular double-stranded DNA that lack histone proteins. So histones are some basic proteins that helps in the packaging of DNA. However, histones are absent in the DNA of prokaryote, because of which prokaryotic DNA or nucleus is called as naked, that lacks histones. Plus, this genetic material is directly diffused in the cytoplasm. Why? Because it is not covered by any nuclear membrane. Then the genetic material, that is the DNA, that is this chromosome. Either you use the term genetic material, or you say DNA, or you say chromosome, one and the same thing. So it is not only naked in terms of not having histone protein, but also it is not surrounded by any nuclear membrane. So genetic material, you write, is directly beta diffused in the cytoplasm. Why? It is directly diffused in the cytoplasm as it lacks membrane around it. There are no nuclear membrane. Rather, when you see a prokaryotic cell like this, suppose this is my prokaryotic cell which has the outermost boundary by the name cell envelope. Cell envelope encloses the fluid which is called as cytoplasm. The cytoplasm is also referred to as the main arena for all the cellular activity. It is the main arena for all the beta cellular activities. Now, inside this cytoplasm, you can see a double-stranded circular single DNA which is completely naked. Naked means no histone proteins plus no membrane around it. I hope till here I am absolutely clear to everyone, right? Plus, the cytoplasm do not show compartmentalization, which means it does not have any cell organelles. So, no, you can write in prokaryotic cell, there are no membrane bound cell organelles reported. No membrane bound cell organelles are reported over here. The only cell organelle which you can see inside them are the non-membrane bound ribosomes. So they just have non-membrane bound 70S type of ribosome. No membrane bound cell organelle. The only cell organelle if you see in case of prokaryote is ribosome, as they are non-membrane bound, and the nature of the ribosome is 70S type. I hope all these points are clear. Now beta, apart from the main genetic material inside the cytoplasm of the prokaryotic cell only, you have some additional genetic material. Same nature, double-stranded, circular. This extra chromosome in the cytoplasm of the prokaryotic cell is known as plasmid. Was it known as? It is known as plasmids. These plasmids, they give extra phenotypic benefit to the bacteria. Maybe, for example, if it is R type of wait. Hello. I hope now I am properly audible. Yes or no? Am I audible now to everyone? So now let's just continue basically what I was telling you. This naked DNA, rather calling it again and again as primitive nucleus, you can use a new term as genophore or nucleoid, right? So these are the few terms, a few information that you have to remember for a prokaryotic cell. Now let's get into this. What is cell envelope? That means first we are going to discuss about, superficially you have studied about the prokaryotic cell, but now over here, very good beta, if again I am audible. Cell envelope and its modification. So now basically I am going to tell you about the boundaries of a prokaryotic cell. We just wrote cell envelope, right? So now we are going to discuss the three important boundaries of the prokaryotic cell. From outer to inner, if we arrange cell envelope, then the outermost is known as Glycocalyx, which is beta followed by cell wall, and then the innermost is known as cell membrane. So these three are the wall layers of any prokaryotic cell. All the three wall layers beta, they together act as a protective unit. If I see all the three layers together, then their main function is to protect the bacteria from inside. So this is their main function. However, if individually I talk about each layer, first starting with Glycocalyx. So Glycocalyx is the outermost layer, which can be of two types. If Glycocalyx is present in the form of thick, tough, resistant layer, then that Glycocalyx is called as capsule. However, Glycocalyx can also be present in the form of smooth, slimy layer, mucilaginous layer, then it is called as slime. Now let's read it from your textbook. Most prokaryotic cell, particularly the bacterial cells, have a chemically complex cell envelope. Complex, why? Because it is represented by 3 layers, tightly bounded by 3 layers. The outermost glycocalyx, then cell wall, and then the inner membrane. सबके पास अपना अपना function है, but together they act as a single protective unit, right? Now, in fact, on the basis of the nature of cell envelope, Christian Gram, he performed a Gram staining experiment and he divided bacteria into 2 categories as Gram positive and Gram negative. So same thing is written over here. Bacteria can further be classified into 2 groups on the basis of the nature of cell envelope and how they are arranged as Gram positive and Gram negative. So Gram negative are those bacteria who at the first instant do not accept the color of the stain, and Gram positive are those who immediately after coming in contact with crystal violet dye, they become blue in color. So who accepts the stain color are Gram positive, and those who do not accept the stain color are called as Gram negative bacteria. I hope it is clear till here. Yes or no? Okay, further. Now, glycocalyx. Now, let's start with each layer one by one. So, first starting with glycocalyx. As you all know, it is the outermost layer. Now, it can be present in 2 forms. It can differ in composition and thickness among different bacteria. Like, in some bacteria, glycocalyx can be present in the form of capsule, or it can be present in the form of thin mucilaginous slime layer. So it could be loose sheath called slime, or it could be present in the form of thick tuff called as capsule. Now, just remember beta, if glycocalyx is present in the form of capsule, then it provides additional function to bacteria, that is protection against the host immune system. However, if glycocalyx is present in the form of slime layer, then apart from protection, it also avoids dehydration of the bacteria. So you can write over here, slime layer protects the bacteria against dehydration, means loss of water नहीं होने देता, dehydration. Similarly, कैप्सूल अगर है तो कैसे प्रोटेक्ट करता है? प्रोटेक्ट्स द बैक्टीरिया फ्रॉम द होस्ट इम्यून सिस्टम, प्रोटेक्ट्स द बैक्टीरिया फ्रॉम द होस्ट immune system. Correct? Yes or no? I hope it is clear to everyone. Now, after talking about the glycocalyx, let's come to the middle layer. The middle layer, which is known as cell wall. Now, basically, beta, cell wall, you know, it is a dead layer and it is completely fully permeable, which means it will allow, I'm here for practicing English. Acha, that's good, Sagar. If you are practicing English in botany class, I mean, good for you. Very good, Sagar. So now what I am saying over here is that now let's talk about cell wall. So cell wall is the middle layer which is dead, fully permeable. When I say fully permeable, I mean to say it will allow any material to pass through it in and out. Now what is the main function of cell wall? It regulates the shape of the cell and it prevents the bursting of the cell. This is the most important function of the cell wall. Apart from providing rigidity, strength and support to the cell, उसके अलावा cell wall का सबसे important function क्या है? To protect the cell from bursting, from collapsing. So just remember this important function of the cell wall. Now let's talk about the innermost layer, which is cell membrane. So when I talk about cell membrane, beta, you can see in the theory as well that I have written on the board, cell membrane is the innermost layer. Unlike cell wall, it is a living layer which is selectively permeable, that it allows only selected molecules and ions to move inside or outside the cell. The structure of plasma membrane was decoded by Singer and Nicholson in the year 1972. Now remember one very important point over here. If you are listening, please note it down somewhere, that the only structure similarity between the eukaryotic cell and the prokaryotic cell is cell membrane. Prokaryote, eukaryote में कोई भी similarity नहीं है when it comes to the structure except cell membrane. Be it cell membrane of prokaryote, be it the cell membrane of eukaryote, both are same, made up of lipid bilayer and protein. सिंगर-निकोल्सन का जो मॉडल था, फ्लूइड मोज़ेक मॉडल, that is applicable to both prokaryotes and eukaryotes. I hope I am clear till here. So the plasma membrane is selectively permeable in nature and interacts with the outside world. That means it allows only selected molecules to move in and out of the cell. This membrane is similar structurally to that of eukaryotes. See, written na? So if you're attending this lecture, so let me tell you, you are not going to miss even a single point of your NCERT. All the points of NCERT will be covered in this lecture fully in detail. So yes, if you are attending, don't think that your time will be wasted. It is always going to be a utilization of your time. A special, now this plasma membrane, if you see in case of prokaryotes beta, since it is living, so it can grow inward as well as outward. So in case of plasma membrane beta, in case of prokaryote, this plasma membrane can grow inward as well as it can grow outward. So if plasma membrane grows inward, it can grow either in the form of lamella or in the form of vesicle or in the form of lamella is this, in the form of tubes, in the form of vesicle or in the form of branched lamellas. In whatever mode plasma membrane grows inward in case of prokaryote, these structures, they are known as mesosomes. So when plasma membrane in prokaryote grows inward in either of the shape, then this additional structure is known as mesosomes. These mesosomes beta, they carry respiratory enzymes. And these mesosomes, because of the presence of respiratory enzyme, basically they can function like mitochondria. That means the function of breakdown of glucose and generating ATP in bacteria is the role of mesosomes. So it's written over here, a special membranous structure is the mesosome, which is formed by the inward extension of the plasma membrane inside the cell. They can be either in the form of vesicles, tubules or lamella. Correct? I hope things are absolutely clear to everyone. Now, apart from this beta, plasma membrane can also form 3 more additional structures by the name flagella, pili and fimbriae. So these are the 3 additional structures which a plasma membrane can form. Flagella are the locomotory organs. Any bacteria with flagella are motile. Pili, they help in the exchange of genetic material between the 2 cells. So pili, they help in sexual recombination. That means exchange of genetic material. Similarly, when I talk about fimbriae, again these are the extensions of plasma membrane which help in the attachment of the bacteria to the substratum. So fimbriae helps in the attachment. Correct? So that only over here it is written. Now bacteria cell may be motile or non-motile. If it has flagella, motile. If it does not has flagella, it is non-motile. Now they have thin filamentous extensions from the wall called flagella. Bacteria shows arrangement. Now bacteria can have 1 flagella or it can have many flagella. Typically, if I talk about the structure of a flagella beta, it consists of 3 parts, that is filament, hook and basal body. Apart from flagella, as I told you, pili and fimbriae are also the surface structures that are extended from the plasma membrane. They do not have any role in motility, but you can see what are their actual roles. That pili help in the exchange of genetic material and fimbriae, they help in the attachment of the bacteria to the substratum. Yes, correct Siddhi. Flagella movement में मदद करता है, pili 2 bacteria के बीच में exchange of genetic material करवाता है and fimbriae helps in the attachment. So that only over here is written. You can read it once. Now, this is all about the cell wall, cell envelope. I hope it is crystal clear to everyone. Now, let's talk about inside the cytoplasm what all other things are present apart from the genophore, apart from the nucleoid in a prokaryotic cell. So, basically what all extra things you can see? Ribosomes and inclusion bodies. See, here itself I mentioned that in the cytoplasm of the prokaryotic cell, it is very simple. Not much complication is present. Why? Because the cytoplasm of prokaryotes, they do not show any kind of, compartmentalization, no membrane bound cell organelles, no ER, Golgi bodies, nothing. Very simple and continuous cytoplasm is found in case of bacteria, except they just have one non-membrane bound cell organelle by the name ribosomes. And apart from ribosome, they have some storage bodies, some dead storage bodies by the name inclusion bodies. ठीक है, तो ज्यादा कुछ नहीं है इनके पास. इनका साइटोप्लाज्म बहुत ही ज्यादा स्मूथ होता है, with not so much of compartmentalization or complexity. So now let's talk about the ribosomes and inclusion bodies. In prokaryotes, ribosomes are associated with the plasma membrane of the cell. See, prokaryotes can be in prokaryotic cell, ribosomes can be found in two places, like I have drawn in the diagram, freely floating in the cytoplasm or attached to the plasma membrane. Correct? Now, they are about 15 to 20 nanometer in size, very, very important. These granular particles, these ribosomes, what is their standard size? They are 15 by 20. Ma'am, when you will take the next class? Why, Siddhi? First, at least you pay attention to this class which is going on, and then otherwise every Saturday I'm going to come, and one lecture of class 11th, another lecture of class 12th. That is how we are going to continue. So next week Saturday, Siddhi, we will do Principles of Inheritance and Variation, the chapter from class 12th, right? Chalo. So shall I continue? Now, see, so what is the standard size of any ribosome? It is 15 by 15 by 20 nanometers. And now you remember that ribosomes, they are made up of two subunits. The larger subunits, now if I talk about ribosomes, just pay attention over here, beta. Ribosomes, they are made up of two subunits. The larger subunit in case of prokaryote is known as 50S, and the smaller subunit is known as 30S. These subunits of ribosomes are actually made up of different types of rRNA and proteins. So if question comes, what these ribosomes units are made up of? So they are made up of rRNA and proteins. Correct? Now 50 is the larger and 30 is the smaller. So same thing is written over here. They are made up of two subunits, 50 and 30, which when present together forms the 70S. So never add 50 plus 30 will give you the sum of 80. However, we say it as 70 because we don't add the numbers 50 and 30. It's all on the basis of the density gradient. So whatever additive calculative density comes, accordingly those numbers comes. 50, 30 doesn't make 80 over here because it is not an additive quantity. Just remember this thing, right? To form 70S prokaryotic ribosomes. Now, what are the functions of ribosomes? So basically, beta, ribosomes are protein factories. They are responsible for the synthesis of protein. Now, question comes, can ribosomes alone produce protein? No. So in the cytoplasm of the cell, these ribosomes, they associate with mRNA to form the structure called polysome, also called as polyribosome, that helps to translate mRNA into protein. So since mRNA uses ribosome for translation, that is why ribosomes are known as protein factories. So ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and forms a chain called as polyribosomes or polysome. I hope all these points are clear. Now last is inclusion bodies. Inclusion bodies. Now what are these inclusion bodies? I told you these are dead non-membrane bound storage bodies which are scattered in the cytoplasm of the prokaryotic cell. And since they are storage bodies, so some are sulfur granules, phosphate granules. So sulfur is stored in sulfur granules, phosphate is stored in phosphate granules. So all storage bodies. Some reserve material is stored in these inclusion bodies. That's it. The one of the inclusion body which I would like to mention again over here is gas vacuole, which are exclusively found in some green algae and cyanobacteria, some blue-green algae and some purple and green sulfur bacteria. They have gas vacuole. They are again non-membrane bound structures which are responsible for trapping gas in them, so that these bacteria, they become light in weight and they simply float on the surface of water. So see over here, inclusion bodies are the reserve material in prokaryotic cell which act as a storage bodies in the cytoplasm. They are not bounded by any membrane system and they lie free in the cytoplasm. For example, phosphate granules, cyanophycean granules, sulfur granules, etc. One of the inclusion body that is mentioned again over here is about gas vacuole, which is found in case of cyanobacteria and purple-green photosynthetic bacteria, responsible for providing buoyancy to the bacteria. I hope all these points are crystal clear, and I hope eukaryotic, prokaryotic cell is again very clear to each and every one of you. Now let's continue and let's discuss about beta eukaryotic cells. Now I told you there are two types of cell, prokaryote and eukaryote. So one you have done prokaryotic cell. Now second is eukaryotic cell. So before I start discussing about this diagram, first let's discuss one thing about the eukaryotic cell, that is the basic points that differentiates prokaryotes from eukaryotes. So now I'm going to give you the heading of eukaryotic cell. Now like you have seen, I have divided pro as primitive, karyo means nucleus. So similarly over here also we can say beta, U means advance, karyo means nucleus. So basically beta, eukaryotic cells are those advanced cells which have well developed nucleus, unlike you have seen in case of prokaryotic cells. So these are those cells which have beta well developed nucleus. Like for example, plants, animals, fungi, algae, protisteans, every organism except bacteria are the eukaryotic organism. सिर्फ बैक्टीरिया को अगर छोड़ दो तो जितने भी organisms हैं, they all fall in the category of eukaryotic cell. Correct? Every falls in the category of eukaryotic cell. I hope this much is clear to everyone. Now, what you mean when we say well developed nucleus? क्या मतलब होता है जब हम कहते हैं well developed nucleus? So, well developed nucleus says that nucleus, that is genetic material. It is good to write genetic material. The genetic material is not diffused in the cytoplasm, rather the genetic material is enclosed within a membrane, right Chandu? So it is enclosed within a membrane and the genetic material is represented by many number of chromosomes, not just one, which are not circular, rather they are linear, and they have the presence of histone proteins along with non-histone proteins, right? So what you will write over here, that is genetic material is not naked, as they have histones with them along with non-histone proteins, plus nuclear membrane is present, plus one more point, genetic material is represented by double-stranded but not circular linear chromosome which are more in number, not just one, which are more than one. As you have seen in prokaryotes, just one single circular DNA, yes or no? But if I talk about eukaryotic cell nucleus, they have more than one chromosome. I hope things are clear till here. Okay. Now, like you have seen in case of prokaryotic cell beta, they lack the presence of membrane-bound cell organelles. There is no compartmentalization in the cytoplasm of the prokaryotic cell. But if you see the eukaryotic cell, I will teach you, you will also see that they have the compartmentalization system. All the membrane bound, be it single, double, all such kinds of cell organelles are found in their cytoplasm. So next point you can write is membrane bound organelles found. But like you have seen in case of prokaryotes beta, plasmid was present, organelles found. The extra genetic material was found in case of bacteria. However, in eukaryotic cells, just make sure there are no plasmids. Why? Because we don't need that extra genetic material. We already have so many chromosomes that plasmid function will be done by our chromosome itself. But you have seen in case of prokaryote, only one chromosome na, so they might need that extra chromosome. So prokaryotes have plasmids, but we eukaryotic cells, we do not have any plasmid. So I hope things are clear till here. Yes or no? Now proceeding further, beta, I told you that in case of prokaryotes, cytoplasm was there as the main arena for all the cellular activity. So same point will come over here also. Even in eukaryotic cell, cytoplasm is found that act as the main arena for all the cellular activity. Now the only difference between the prokaryotic and the eukaryotic cytoplasm is, in prokaryotic cytoplasm they do not show movement, they do not show cyclosis, but here in eukaryotic cell, beta, the cytoplasm, the fluid, will show the circular motion. In our cell, if you study it under the microscope, you will see cytoplasm is not constant, it keeps on moving. So you can say cytoplasm in our cells shows circular motion, which is not the feature of prokaryotic cell. Plus, there you have seen the presence of 7. Thank you, Chandu. I have already replied. Thank you so much, beta. Thank you. Yes, I know I am cute. Just come up with something new, so that I also get mesmerized. Right, Chandu? I know. Thank you so much, beta. Okay. Hello, bhai. Hello. Now, shall we continue? Thank you, Maithili. Come up with something new, guys. I know I am cute. Tell me something new just to mesmerize me, right? So cytoplasm shows circular motion. Now there you have seen 70S type of ribosome, but here you will see the presence of beta 80S type of ribosome in their cytoplasm. So in the cytoplasm, you will report the presence of 80S type of ribosome. 80S means again 2 subunit. The larger one is called as 60S and the smaller one will be called as 40S. They are made up of they are made up of rRNA and protein. Same size, which is 15 by 20 nanometer. I hope I am clear till here. Yes or no? If yes, then let's continue with the structure of eukaryotic cell. Basic features you all have studied. The basic difference between prokaryotic and eukaryotic cell. Now it's time, like you have seen the structural aspect of prokaryotic cell. Now it's the time to tell you the structural aspect of a eukaryotic cell also. Now beta, let's talk about the cell envelope. We have discussed the prokaryotic cell envelope, right? Now here we are going to start the discussion of the cell envelope of eukaryotic cell. So basically beta, let me tell you one thing, that majorly there are 2 types of eukaryotic cell, the plant cell and the animal cell. In animal cells, Theodor Schwann already mentioned that cell envelope is only represented by plasma membrane, whereas in case of plant cells, there are 2 layers of cell envelope. The inner one is cell membrane and the outer one is cell wall. So in any eukaryotic cell, we have 2 layers. The common one between both plant and animal is cell membrane, which is the inner one, and the exclusive one for plant cell is the cell wall. So first, we are going to start with the discussion of the plasma membrane structure. Correct? Yes or no? Now, just 10 minutes back, when I was teaching you the prokaryotic plasma membrane, there I mentioned one thing. Can you really see, Chandu, my eyes? I am wearing these big glasses. Can you really see my eyes? Huh? No. If I'll remove the specs, you will see the dark circles of my eyes. Now, do you really find my eyes beautiful? Okay, fine. So if I talk about the beta plasma membrane, so plasma membrane is the only structure between prokaryote and eukaryote which are same. Either you study the structural and the functional aspect of the prokaryotic plasma membrane, or you study the plasma membrane of eukaryote. It is absolutely same, and this is the only structural similarity between the 2. Otherwise, all other structures are exclusively different, right? So what you will write over here? Plasma membrane. Both prokaryote and eukaryotes have same structure and function of plasma membrane and function of plasma membrane. That is, plasma membrane is living, selectively permeable and allows bulk transport across the cell. So it is the main plasma membrane which interacts with the outside world. Thank you. So yes, you must have, there's a very old saying in Hindi I recall, Jo humse takrayega, now you people complete it, right? So it's just like the same, the same thing I follows. Who tries to mess with me, I'll reply in the same manner. Okay, so it was just a healthy roast. I hope this Chandu, my lovely student, is not going to get annoyed. So it was just for the sake of fun. Chalo, okay. So shall I continue? So basically, plasma membrane is living. You have already studied. It is selectively permeable. I have already told you the meaning of being selectively permeable. And bulk transport. So bulk transport simply means that this plasma membrane is actually responsible for contacting from the outside world. That means from outside, whatever is going to enter inside the cell or vice versa, everything is going to be controlled by this plasma membrane. I hope till here everything is clear. Now let's proceed with the structure of plasma membrane. The most accepted model of explaining the structure of plasma membrane was given by Singer and Nicholson in the year 1972 by the name fluid mosaic model. This diagram I have picked up from your NCERT. This diagram is very, very important. All these labelings that you see over here are absolutely very, very important. So just make sure that you are by heart with this diagram as well as the important points. So the best model was proposed by Singer and Nicholson while studying on the human RBC. So when by 1972, ultra electron microscopes, everything was discovered, everything was invented. So it became very easy for Singer and Nicholson to have a view of all the structural details inside the cell. So they got an opportunity to study the structure of plasma membrane in well detail. So they chose which eukaryotic cell? They chose human RBC. And what they found that plasma membrane is represented by two major chemical, lipid and protein. The main skeleton is made up of, ma'am, what is fluid mosaic model? The best definition. Okay, Anuj, fluid mosaic model was the model that explains the structure of plasma membrane. It was proposed by Singer and Nicholson in the year 1972. First 2 points, where fluid represents your lipid bilayer and mosaic represents the proteins embedded on it. So fluid is the lipid bilayer, mosaic is the protein on that fluid. It's like there's one line written in your NCERT for fluid mosaic. It seems like proteins are floating in the sea of lipid. Matlab, fluid is the lipid on which proteins are present here and there, and they can show movement on the protein. GN Ramachandra plot is not required, Krishna. Let me tell you, the little bit information is given in the first as a scientist, because when you start this chapter, Cell the Unit of Life, the description of the scientist is of GN Ramachandra, who is famous for his plot, that graph. But at your level, it is not at all required. It is a very depth graph. It is a very depth explanation, which at your level to understand is not that very easy. And I have never ever seen any question coming in detail from this graph. It's important that you just learn the name of the scientist and his contribution from that just one paragraph. Okay. I hope Anuj I am clear, and Krishna I hope I am clear to you as well. So now can I continue? Okay, so now let's talk about this lipid bilayer, the model which was proposed by the model which was proposed by Singer and Nicholson. So I'll draw my diagram also. So as Singer and Nicholson said that the plasma membrane is represented by two layers. The upper layer made up of lipid, the lower layer also made up of lipid. Now this lipid is majorly associated with phosphate. Therefore we say phospholipid. Yes ma'am, in NCERT fluid behaves like liquid which floats in the membrane, and mosaic is like tiles. Yes, that's correct. No, fluid, I'm too confused by, see Anuj, correct, you are saying it right. Fluid means what? Liquid. So here in this model of lipid bilayer and protein, what represents fluid part? The lipid part. And what represents the mosaic part? Mosaic is represented by the proteins which are present here and there on the lipid bilayer, and that can show movement. It's like proteins are the iceberg which floats in the sea of lipid. Correct? Okay, now shall I continue? So basically we have two layers of the lipid, the upper and the lower. Remember beta, these lipids are mostly associated with phosphate. Therefore, they are also called as phospholipid. Now, I'll take out one molecule of the lipid to make you people understand. This circle is called as head, and this tail, this is the tail part. Remember, head is made up of saturated hydrocarbon, which are hydrophilic in nature. But tails are made up of, the tail is made up of unsaturated hydro. Ma'am, but teacher, my answer, when I did not understand. Anuj crosses your answer when I wrote this. What you have written? You have written correct. There's nothing wrong. Maybe your teacher must be expecting from you that you should mention, because what you have written in the message, from there you haven't mentioned that what is fluid and what is mosaic. Fluid is liquid, but which liquid? So you have to mention lipid. Mosaic is the tile, but which tile? Protein. That till the time you won't mention these two terms, you won't get the answer. You might be right in your explanation, but till the time you do not mention that what fluid stands for, what mosaic stands for, you're not going to fetch the marks. Correct, Anuj? Okay, so head is hydrophilic in nature, whereas tail is beta hydrophobic in nature. Always remember that outside the cell and inside the cell, it is all aqueous environment. Yes or no? Ma'am, what about that? We have done beta. Sexual reproduction in flowering plants, we have done, bache. You can, till apomixis, I remember we completed all the topics. Okay, so here is also the aqueous environment. Here is also the aqueous environment. So now, since tail is hydrophobic and does not like, okay, thank you Anuj. So aqueous environment. Now since it's aqueous environment inside and outside, therefore this head is hydrophilic in nature. Therefore it can come in direct contact with the inside and outside environment of the cell. However, tail being hydrophobic, it is always protected by the head. So you're always going to project tail inside and head outside. This is how the lipid bilayer has to be arranged. And now on this lipid bilayer, beta, proteins are found. There can be two types of proteins, either completely surface protein, means extrinsic protein, or intrinsic proteins that are embedded inside the membrane. Now always remember, you can see over here in the diagram, see this is one layer, this is another layer of the lipid which is mentioned as phospholipid bilayer, on which you can see proteins are present. See, some are completely peripheral protein like this. If you can see it in the diagram, they are only present on the surface. These are peripheral proteins which are only present on the surface. But here if you see, this is the integral protein, the protein which crosses the membrane, which is present inside also and outside also, right? So there are two types of protein, intrinsic and extrinsic. Now this intrinsic protein, if it wants, it can show lateral movement across the lipid bilayer. Lateral means like this, not upside movement, not flip-flop movement, only lateral movement. So it seems like this protein as an iceberg is floating in the sea of लिपिड। ऐसा लगता है तैर रहा है क्योंकि अपनी पोजीशन पे फिक्स्ड नहीं है. इट कैन मूव लेफ्ट एंड राइट बट नॉट अप एंड डाउन. I hope it is clear to everyone. So now let's continue. And so apart from this, you can also see that on this lipid bilayer, on the extrinsic protein, you have sugar molecules also attached. ठीक है? So sugar molecules can be directly attached to the lipid as well, or it can be attached to the peripheral protein as well. Apart from lipid bilayer and protein, plasma membrane also has cholesterol. So it also has cholesterol molecules, right? Anuj ma'am, explain fluid again. I'm too okay. Fine, Anuj. We will do it again. Yes, ma'am. But I always get while reading the plasma. No, there's no point. No, there's no point of getting confused again and again. So always. So now let me write it for you all. Fluid mosaic model, where fluid means what? Fluid means lipid bilayer. And mosaic is represented by protein, right? So fluid is represented by lipid bilayer, whereas mosaic is represented by protein, where we say that proteins shows, especially intrinsic protein shows lateral movement on the lipid bilayer. Very, very simple. Yes or no? The structure and the scientists are clear. I hope now, Anuj, it must have made the sense to you that what is lipid bilayer and what is the role of the protein. So lipid bilayer is just the bilayer on which proteins are present, and it is the function of the protein which helps the movement of material in and out of the cell. Like for example, through this integral protein, basically beta, what can be done through this integral protein? Any molecule or ion can be made to pass, right? I hope till here it's clear. So this is about the structure of plasma membrane. So plasma membrane, they are living, selectively permeable. Their main function is to allow the bulk transport. The best model was proposed by Singer and Nicholson in the year 1972 by the name fluid mosaic model, while studying on the eukaryotic RBC cells, where they found that the RBC plasma membrane is made up of 40% lipid and 52% protein. Rest are cholesterol and etc. Okay, now what is this lipid bilayer? This is how the lipid bilayer has to be present, which is associated majorly with phosphate. Hence we say phospholipid bilayer. Bi matlab दो layer हैं, एक ऊपर की एक नीचे की. The lipid simple molecule I have taken out and I have shown you that it is represented by hydrophilic head and a hydrophobic tail. This hydrophilic head, it always faces outside. Why? Because outside is the aqueous environment, so head has to be outside. That protects the tail inside. Correct? As tail is hydrophobic in nature. अब इस पूरी lipid bilayer पर क्या present होते हैं? Proteins, which can be of two type, either present on the surface or embedded inside the membrane. If they are present on the surface, they are easy to remove and they are called as peripheral protein or extrinsic protein. You can see it in the diagram also. Keep comparing. Or the integral proteins, as you can see, the intrinsic protein which crosses the membrane and majorly helps in the movement of material. These integral proteins, they are not fixed in their place, right? These are not fixed in their place. They can show lateral movement. Lateral matlab sidewise, not up and down. Sidewise movement they can show. That is why we say that in the sea of lipid, proteins float as iceberg. लिपिड के समुंदर में प्रोटीन बर्फ के टुकड़े की तरह फ्लोट करते हैं लेफ्ट राइट. करेक्ट? सो हियर फ्लूइड मींस लिपिड, mosaic means protein, because on the sea of lipid you can see protein here and there. I hope it makes the sense to everyone. Now let's continue further, and now let's talk about the second layer, which is cell wall. Now Anuj and Pawan, I hope now plasma membrane makes sense to you. Yes or no? Is it clear? Thank you, Ruchi. Now does it make sense to you? If yes, then we can continue. Now, cell wall. So remember beta, animal cells do not have cell wall. Cell wall is found only in some eukaryotic cells like plants, algae and fungi. Now, the main function of cell wall is like it's dead, it's fully permeable. So the main function is to prevent the cell from bursting. It gives shape to the cell, as well as it also provides rigidity and strength to the cell. Now, depending upon what cell wall is made up of, we have 3 different types of cell wall. Everything I'm going to read it from here, from your NCERT. I hope NCERT is open in front of you all. As you may recall, a non-living, I told you, a dead layer. Means what? No. No, they are just complimenting me. So, Anuj, yeah. I should wash my face. So, is it just a compliment or is it just a comment? I'm not able to understand, Anuj, that why should I wash my face? Will washing my face, will that enhance my beauty? Or it will open their eye? Will it be like an eye opener that, see, ma'am is not that beautiful? Or it will enhance my beauty? What do you think, Anuj? Should I really wash my face, or am I still looking pretty to you? Okay, so as you may recall, a non-living rigid structure, and okay, washing my face doesn't mean that I have put makeup. No makeup. So I'm completely natural in front of you. Yeah, that's true. It's okay, don't say sorry. Kidding, yaar, come on. See, we have this one short video which cannot be very, ma'am, please teach me how to speak fluent English. Ruchi, sorry beta, I'm not in English class. This may be an English platform, but I'm so sorry, I'm not your English teacher on this English platform. I'm the biology teacher, who even I'm not that fluent in speaking English. I can only speak English when it comes to biology. So yeah. Okay, thank you beta. So basically, if I talk about the cell wall, it is a non-living dead layer, rigid layer, and they are present outside the plasma membrane of fungi and plants and algae. Cell wall not only gives shape to the cell, but it also protects the cell from any kind of damage and infection. So cell wall is the outermost layer, which is again dead, fully permeable, mainly providing strength and rigidity to the cell and not letting any kind of damage coming inside the cell. It maintains the cell shape, etc. So these are the basic functions. And now, depending upon the composition, as I told you, so just underline, very, very important. Question can be put up from this segment. Remember, algal cell wall exclusively, apart from cellulose, it also have some other sugars like galactans, mannans, and may also have crystals like calcium carbonate. Plant cell wall exclusively made up of cellulose, hemicellulose, and pectin. Fungal cell wall is made up of chitin, which is the homopolymer of NAG. So all this is written in front of you. You may read it from your NCERT. Now beta, cell wall, depending upon the origin, can be of 2 types. Primary cell wall and secondary cell wall. Remember beta, primary cell wall is present around the young plant cell, which is thin in nature, flexible, and can grow. With time, when that immature young plant cell grows, matures and becomes mature plant cell, the primary cell wall expands and new secondary cell walls are laid inner to it. Why inner? Because intrinsic growth is the feature of living organism. तो प्राइमरी सेल वॉल एक्सपैंड करेगी और उसके अंदर कई सारी सेकेंडरी सेल वॉल्स आएंगी. Now remember, primary cell wall was living in nature, capable of growth, thin and flexible. But when it grows and when new secondary cell wall comes, they are thick in nature, they are dead in nature, they are not flexible, they cannot grow further. Yes or no? I hope I am clear. So primary cell wall is capable of growth, which is found in young cell, which gradually diminishes. Primary cell wall समय के साथ चली जाती है. Why? Because it expands, it breaks, and new secondary cell walls inner to it are laid down as the cell matures, which is dead in nature, that is non-living, not made up of, not capable of growing, that is, it is not flexible, not capable of expanding. I hope it's clear till here. Now, like you see, if I want to make a wall, so I have to lay bricks, and in between the bricks, I have to lay a cement, right? So that those bricks, they stick together and they make a strong wall. So similarly, if my body, if I talk about plant, because we are studying botany, so my main focus is going to be on plant cells, plant body. So if I talk about a plant body, it is made up of large number of cells. Now all cells, they have to be glued together now, otherwise the body is going to be very loose. So that for stiffness, for that strength in the plant body, all the cells need to be connected, and for that connection, what is present between the two cells? It is the middle lamella. So middle lamella is actually the chemical which glues the two cells together like a cement, and it is majorly made up of calcium magnesium pectate. So the middle lamella is a layer which is mainly made up of calcium magnesium pectate. Its main function is to hold the two cells together. But now if the two cells, they are packed with a cement, then how the two cells are going to contact? So for that, through the middle lamella, a plasmodesmata is laid down, which is a living connection between the two plant cells, so that the two plant cells can communicate and can allow the exchange of cytoplasmic content. So they are traversed by plasmodesmata in order to connect with each other. I hope till here all the things are crystal clear. Now this is about the plasma membrane and the cell wall. Now let's come inside the cytoplasm. As I told you, unlike prokaryotic cell, inside the eukaryotic cell, basically beta, you see compartmentalization in the form of presence of cell organelle. So first I am going to list you 4 types of cell organelle that makes the concept of endomembrane system. So now we are coming inside the eukaryotic cell, be it a plant cell, be it an animal cell, they all have this endomembrane system. Endomembrane system is represented by 4 such structures which are single membrane bound, and all the 4 are functionally coordinated. So the 4 functionally coordinated single membrane bound structures are ER, followed by Golgi, followed by lysosomes, and then finally vacuoles. So these all 4 are, first point, they are single membrane. Matlab around them how many layers is present? Only 1. Second, they all are functionally coordinated. They are single membrane bound and all are functionally coordinated. Now let's try to understand that how all these 4 are coordinated. Now I'll try to explain all these things with the help of a diagram. ठीक है, एक डायग्राम की मदद से हम पूरी कहानी को बेटा समझने की कोशिश करते हैं. So suppose this is my cell, this is my eukaryotic cell, and suppose a eukaryotic cell will have a well developed nucleus, right? So suppose here on little bit over here I'm trying to draw the nucleus. However, the nucleus in case of animal cell is always central. So here I'm drawing the nucleus which is double membrane bound. Now why am I drawing the nucleus? Any guesses from anyone? I have nowhere mentioned that in the endomembrane system that any of the organelle involved is nucleus. The 4 cell organelles are ER, Golgi, Lyso and Vacuole. But when I am trying to start the explanation, I am starting with drawing a nucleus. Why? So let me tell you one thing beta, because the outer membrane of the nucleus is the cell organelle which is the attachment site for ER. So whenever I have to start the endomembrane system, that means whenever I have to start my concept of endomembrane system and whenever I have to start with ER, एंडोप्लाज्मिक रेटिकुलम, जब भी मुझे इससे शुरुआत करनी होगी तो सबसे पहले मुझे क्या बनाना होगा? द फर्स्ट थिंग विच आई नीड टू ड्रॉ इज द nucleus. Why? Because it is the outer membrane of the nucleus which gives the attachment site to the ER. Now structurally, first let me tell you, structurally ER is represented by 3 parts. The flattened sac-like structures which you see over there are called as cisternae. Cisternae may give rise to tubule-like structures. Tube-like structures called as tubules. So cisternae are flattened sac-like structures interconnected to each other, which may fold to form tube-like structures, tubules, and finally they may give rise to ball-like structures called as vesicles. So ER is typically represented by 3 parts. Now there are 2 types of ER depending upon the function: smooth endoplasmic reticulum and rough endoplasmic reticulum. So the ER on whose cisterna you see the presence of granular bodies, that is ribosome. So these are the ribosome. In eukaryotic cell, what is the type of ribosome? It is the ATS type of ribosome. So these ATS type of ribosome, so this ER will now be called as RER. And the type of ER whose cisterna lacks ribosome, that means they are smooth. So they are called as SER. I hope now this much is clear. Now this ER divides the cytoplasm into 2 compartments. The inner space which is present inside the ER, this is called as luminal space. And everything outside the ER is called as extraluminal. Itna yaad rakhoge? Okay. Now beta, if I talk about the function of RER, let's talk about the ER function. Exclusively, RER is responsible for the synthesis of protein and SER is responsible for the synthesis of lipid, correct, and other steroid-based hormones. So basically, beta, whatever ER is synthesizing, be it lipid, be it protein, whatever they are making, it is completely, you can say, raw. Now, before I start giving you a detailed explanation of this endomembrane system, let's try to understand with a very basic example of day-to-day life. You know, the farmers are the one who grow the crops. So farmers are like the ER who are responsible for growing the crop, like maybe wheat, rice, etc. But whatever farmers grow, can you directly buy from the farmer and can you directly use that wheat for cooking the roti? No. Why? Because the food, the crop which is grown by the farmer, it first need to be processed na. उसको पहले तैयार करना पड़ेगा ना. So all that wheat and rice which are produced by the ER, that means farmer, is first loaded in the truck from the khet, that means from the farm. It is loaded in the truck and it is sent to the factories where these are first unloaded, then they are processed. फिर factory में उनकी पूरी processing होगी, कटाई छटाई होगी, and then all that ready wheat and rice, they will be packed up, है ना, and then again they will be loaded in some different trucks and then from the factories they will be dispatched maybe to the same city or outside the city depending upon the requirement. Yes or no? So who is responsible for producing? Farmer. So here also ER is like the farmer who produces the crop, but whatever he produces is completely raw. It cannot be used directly. It needs modification. It needs changes. Therefore, farmer loads the unmodified food, maybe wheat or rice, in the truck so that it can be sent to the factories for modification, है ना, ताकि उसको हम तैयार कर सके. And finally factories, they modifies the product. Then finally what happens? They are again loaded in the truck for dispatch, है ना. Now either in the same city or different city depending upon where that product is required. Either in the same city or different, भाई जैसी जरूरत होगी. So now let's compare this whole algorithm with our own algorithm. So ER is the farmer who produces the actual crop in the form of proteins and lipid, but whatever he produces is completely raw and it is not ready to be utilized. So ER produces proteins and lipid via RER and SER. that are loaded in the truck means in the vesicles. So these are the vesicles which actually act as the truck, that are loaded in the vesicle for the modification, उनको ताकि तैयार कर सके. और कहाँ तैयार करने ले जाते हैं? What is the factory over here? So the factory is actually equivalent to the Golgi bodies. So it is the Golgi bodies whose function is for to modify the raw protein and lipid. Now, once the Golgi body has done that modification job, now the proteins and lipids are ready. That means the product, the food is ready. Now from here again, it will be loaded in the truck. The completely modified ready proteins and lipids again will be loaded in the truck, means in the vesicle that will arise from the Golgi body for dispatch. Now that dispatch can be done either in the same cell or outside the cell, maybe for different cell. Correct? So this is how the all algorithm is. Let's try to understand over here. So RER has synthesized a protein which is now not ready to be utilized. So this will be packed in the vesicle. That means it will be loaded in the truck. This truck will move out from here. It will come in the cytoplasm. Now it is going to the factory. And what is the factory? Factory is the concentrically arranged Golgi bodies. So this whole structure is my Golgi. Golgis are also better structurally similar to SER, which has 3 parts. The 3 parts are same: cisterna, the smooth cisterna, tubules and vesicle. The first cisterna to receive the truck, that means the first cisterna to receive the vesicle from the ER is known as the cis cisterna. Now here the protein will be discharged or the lipids will be discharged according to the vesicle whatsoever it is bringing. The modification process will begin. The modification of protein is known as glycosylation, and that of lipid, it is known as glycosidation. Now this modification will continue, and then finally the completely modified lipid or protein will reach to the last phase of the Golgi, which is called as trans phase. इसे क्या कहते हैं? Trans कहते हैं. Now finally the completely modified lipid and protein is now ready to be utilized either within the cell or outside the cell. So again a new vesicle, a new truck will come. This completely modified structure will be loaded. The truck will detach from the Golgi body. Now it can be actually via exocytosis, it can be sent outside the cell if the requirement is outside the cell. However, if the requirement is within the cell, then this vesicle will remain inside the cell. And suppose this vesicle has all the proteins which are capable of forming 40 different types of hydrolytic enzymes, then this is going to detach and it can behave directly as lysosome. So if the question comes, अगर आपसे question पूछा जाता है that what is the role of ER, now you see over here how the structures, how all the 4 structures are interdependent on each other. Role of ER is first, this ER begins the set of function by actually synthesizing protein and lipid. But these proteins and lipids are raw and they need modification. Therefore comes the role of Golgi. So Golgi actually does all the part of modification, followed by packaging and then loading in the truck. That means secretion. So all that part of making the lipid and protein ready to be used either within the cell or outside the cell, all that job is of Golgi. So Golgi will only work when ER has synthesized protein and lipid. Suppose ER hasn't done its work. then will Golgi be able to do anything? Answer is no. Golgi will only be able to function when ER has done its job. So isn't it Golgi dependent on ER? So they are functionally coordinated, yes or no? Now once Golgi has done its function, then comes the next cell organelle, which is lysosomes. Now lysosomes are the again single membrane bound cell organelle which are actually responsible for intracellular digestion. These are the cell organelles beta which contains 40 different types of hydrolytic enzymes. So they have 40 different types of hydrolytic enzymes. Therefore, their role is to allow intracellular digestion. They digest any foreign particle which is not supposed to be present inside the cell. उसको बेटा ये digest करने में मदद करता है. और ये बना कैसे? ये बना है क्योंकि Golgi has done its role. So you say that lysosomes, they are the result of packaging of Golgi body. Correct? So if you see over here, see, so basically this vesicle which comes out from the ribosome, if it contains what? If it contains yes, so now if this contains basically beta hydrolytic enzymes, then this vesicle can directly behave as lysosome. Now suppose this lysosome catches any foreign particle as macrophage. So this macrophage is maybe floating in some intercellular space. So this plasma membrane will bring this macrophage inside and will feed it to the lysosome. So now lysosome, with the help of its 40 different hydrolytic enzyme, what will it do? It will digest the macrophage, the unwanted particle. अब सब कुछ तो डाइजेस्ट हो नहीं जाता, so a part of this macrophage may be digested by the lysosome. Now whatever undigested part of this macrophage remains inside the lysosome goes and gets temporarily stored in the vacuole. So vacuole is just like the dustbin of the cell where all the secretory and excretory products are stored temporarily. जैसे घरों में हम डस्टबिन रख देते हैं ना, and what is the role of that dustbin? The role of the dustbin is just to keep the all those your household waste, the kitchen waste for just a day. Next day morning, as soon as it happens, that kude wala visits and then you throw all that dustbin waste in that truck, the dustbin truck which you see. So basically it's just similarly over here also. This vacuole acts as a dustbin of the cell where temporarily all the undigested or the secretory and excretory products are stored. So whatever lysosome digests, well and good, but if any undigested part is left, so that comes and gets temporarily stored in the vacuole. So you can very clearly see that how all the four membrane bound, single membrane bound structures are functionally interdependent on each other. Yes or no? I hope I'm very much clear to each and every one of you. If everyone has understood till here, then now let's proceed further. So this is about all the four structures. Now little bit let me tell you about lysosome and vacuole. Golgi you have seen, structure is similar to that of SER, and function of Golgi also you have seen, as well as of ER. Now it's time to do something for lysosomes. So lysosomes, they are single membrane bound, which were discovered by scientist Christian D. Dube. Okay, the scientist who discovered ER was Porter, and the scientist who discovered Golgi bodies, by the name itself you can recall Camillo Golgi, and lysosomes, they were discovered by Christian D. Dube. So they were discovered by Christian D. Dube. they contains 40 different types of hydrolytic enzyme like carbohydrases, nucleases, lipases, DNAs, RNAs, all other enzymes which are actually responsible for digesting some or the other chemicals. So 40 different types of digestive enzymes or hydrolytic enzymes. Correct? Now, proceeding further, lysosomes, these enzymes are only active at acidic pH. They're active at acidic pH. Again, a very, very important point that you people should remember. Next, now, these lysosomes, when they do their function, when they do their job, always remember lysosomes can be of 4 different forms. 4 different forms of lysosomes are available as primary lysosome that directly comes from the packaging of Golgi, secondary lysosome, primary lysosome along with the macrophage, then tertiary lysosome, a lysosome that contains undigested food, and then the quaternary, which is also called as suicidal bag. So these are the few important information which you have to remember. Now then coming to the vacuole. Now vacuoles, when you talk about eukaryotic vacuole, unlike that of prokaryotic vacuole beta, these are membrane bound and their membrane is called as tonoplast. This is selectively permeable layer. Always remember, in animal cells, vacuoles are not that well developed. But when we talk about the plant cells, 90% of their total space is actually occupied by vacuole, because of which in plant cells you will see that the cytoplasm and the nucleus is shifted towards the periphery. साइड में आ जाते हैं क्योंकि vacuole ने 90% स्पेस अपनी ऑक्यूपाई कर रखी होती है. करेक्ट? Next point. Now, whatever has to be stored inside the vacuole, so when the movement occurs from the cytoplasm to the vacuole, so this movement is ATP driven. Energy has to be has to be खर्च करना पड़ेगा, मतलब energy has to be lost. Why? Because when you move any chemical from the cytoplasm inside the vacuole, it is always against the concentration gradient. So these are the few important points which you have to remember, and few examples of vacuole which you usually found in case of eukaryotic cells are food vacuole, contractile vacuole. Gas vacuole is for prokaryote, not for eukaryote. Food vacuole, contractile vacuole, and sap vacuole. Sap vacuole is the one which you see in case of plant. Food vacuole is very much common in amoeba. Contractile vacuole is similarly also very common in amoeba. Food vacuole, as the name suggests, it helps in the storage of food. Contractile vacuole helps in osmoregulation, and sap vacuole helps in the storage of excretory and secretory product, especially in case of plant cells. Now, this is all about the endomembrane system. Now, basically beta, moving towards the next, that is your all the double membrane bound cell organelles. First, starting with the mitochondria. Remember beta, mitochondria are the double membrane bound cell organelles which are also called as powerhouse of the cell. Why? Because they are actually responsible for ATP synthesis. How? Because they help in the respiration process. They help in the respiration process. Now, I told you that they are double membrane bound. Correct? So, here in the NCERT diagram itself, you can very clearly make out that there are two membranes. This is the outer membrane, and inner to the outer membrane, you have inner membrane. Okay, here I'll draw my own diagram also, just to make things even more clearer. So you see, this is my outer membrane, and inner membrane, if you make out, it is not smooth, rather it is present in the form of finger-like projections. So this inner membrane and the finger-like projections can also be called as cristae. Correct? Now space inside the cristae, space which is enclosed inside this inner membrane, this is called as matrix, in which you see double-stranded circular DNA and 70S type of ribosome. Now you can make out one point, that this DNA presence in 70S ribosome you have already studied in prokaryotic cell. So there are so many theories by the name endosymbiotic theory which suggest that mitochondria was actually a prokaryotic cell which for the sake of shelter entered inside a eukaryotic cell. In return, it gave energy to the eukaryotic cell. And then from there onwards, it became a symbiotic association between the two. And now in modern day, we call that prokaryotic cell as mitochondria. Correct? So prokaryotics, this mitochondria is nothing but a prokaryotic cell which got an opportunity to enter inside the eukaryotic cell only for the sake of shelter. And why eukaryotic cell gave the shelter? Just to seek energy, because it could synthesize its own energy. I hope everything is crystal clear till here. Yes or no? If yes, then let's proceed further. So this is about the mitochondria, and always remember, mitochondria, they are not very easily visible under the microscope, just like plastids. So to view mitochondria, you have to stain them densely with some specific stains, then only these mitochondria are visible. And the number of mitochondria is also highly variable from cell to cell. It all depends upon that which cell require more energy. Like for example, if I talk about my leg cells, obviously they will require more energy because every day I have to walk, so more energy is required in my leg cells. So maybe my leg cell will have more amount of ATP as compared to maybe my cheek cells. So just giving an example that the number of mitochondria distribution depends upon the requirement of energy. Plus, what is the shape of the mitochondria? They are cylindrical sausage shape. You all must have seen the burger or the hot dog burger, how it is flattened and cylindrical in shape, sausage shape. So that is the exclusive shape of the mitochondria, right? I hope I'm very much clear to everyone. Similar to mitochondria is plastids. Plastids are large in size and they're very easily visible under the microscope, even if you're not using any kind of stains, right? They're large in size as compared to mitochondria. Second, mitochondria is reported in both prokaryotic and eukaryotic cell. But when it comes to plastids, remember, they're only found in plants, euglena, and algae. So plants, euglenoids and algae are the only ones which have plastids. No fungi, no animal cells, no bacteria will have plastid. Now, there are three types of plastids. We can have three interconvertible types of plastids. The three interconvertible forms of plastids are, on the basis of color and the function, chloroplast, chromoplast and leucoplast. All the three forms of plastids are basically beta interconvertible. Chloroplast and leucoplast. Now see how all these three can be interconverted. You must have seen green chilies and red chilies. So when the chili is young, full of chlorophyll, it is green. But as soon as it ripes, its color changes from reddish to orange. That means all the chloroplast have converted into chromoplast. Ma'am, what's ADP? ADP stands for adenosine diphosphate beta. It is the lower state of energy. And if I add one more phosphate group to ADP, it turns out to be ATP, that is adenosine triphosphate, with maximum state of energy. Correct? Yes or no? So I hope things are crystal clear till here. Now let's continue. All the three forms are interconvertible. I told you, if I have a young chili, it will seem to be green. Or if you have an unripe mango, hare rang ka hota hai. But with time, that mango becomes, when it ripes, it becomes yellow. Why? Because all the chloroplast of the cell has changed into chromoplast. So basically, beta, chromoplast are the colorful plastids. Why they are colorful? Because they have certain colorful pigments. So since they have the presence of colorful pigments, they become colorful. And like for example, xanthophyll, viola xanthin, carotenoids, etc., lycopene, the red colored, the orangish colored is lycopene pigment, and their main function is to impart color to the flowers and the fruit to make them attractive, etc. Chloroplast are the green plastids. whose function is to perform photosynthesis. They're exclusively found in the leaf and they're responsible for trapping sunlight and for producing food. Leucoplasts are the colorless plastid whose function is storage of food. Now, food can be stored in three different forms, either in the form of protein, then that is called as aleuroplast form of leucoplast. Either it can be stored in the form of oils, then that is called as elioplast. Or it can be stored in the food can be stored in the form of sugar, then that is known as amyloplast. Correct? Like for example, if I see a potato is colorless from inside, right? Whitish colorless hota hai. Why? Because majorly in the potato cells, the form of plastid is leucoplast, where the food is stored in the form of starch, like amyloplast. So from inside it is colorless. Now typically, beta, in one of the plastid which you have to study in detail is the chloroplast. So here you can see the diagram of the chloroplast, which is double membrane bound like mitochondria. The outer membrane, the inner membrane. The inner membrane encloses the ground substance which is called as stroma. The stroma consists of some coin-shaped structures, individual called as thylakoid, and the piles of these coin-shaped structure. The pile is called as, the heap is called as the grana. This grana system is actually responsible for trapping the sunlight and converting it into chemical form. Correct? So you have grana, you have thylakoid. Each set of grana, you can see how they are interconnected to each others. So these interconnections, they are called as stroma lamella. Now these chloroplasts, the number of chloroplasts in the leaf can vary from 1 to 40, right? And their shapes are also highly variable. Like for example, if I talk about the shape of chloroplast in a unicellular green algae Chlamydomonas, so there it is cup shaped. If I talk about the shape of chloroplast in Spirogyra, so which is a multicellular filamentous green algae, there it is spiral shaped. In Eulothrix, it is collar, girdle shape. So the shape of chloroplast and the number of chloroplast varies from cell to cell. However, its function remains same. What is the function of the plastid? Function of the chloroplast is to trap sunlight and convert it into food, which is glucose. So not only it synthesizes the food, but it can also store the food. I hope till here things are absolutely crystal clear in each and everyone's mind. Chalo. So now let's proceed further. So this is all about the plastid. Now we are moving towards the next topic, which I have already done in case of prokaryotic cell. Still we are going to continue over here, which is ribosomes. So ribosomes, standard size is 15 by 20 nanometers. They are the granular particles which were discovered by George Palade. So they are granular particles which were discovered by George Palade in the year called 1953. As I told you, these ribosomes, they're actually made up of rRNA and protein. They are non-membrane bound, be it 70, be it 80. They are non-membrane bound cell organelle. That is why they are found in case of prokaryotes also. Now in eukaryotic ribosome, the nature is 80S type, but in prokaryotic it is 70S. Now each ribosome has two subunits. The two subunits are the larger one and the smaller one. In case of eukaryote, the larger is 60, the smaller is 40S. Correct? So each ribosome has two subunits, the larger and the smaller. Now the two subunits of 80S are 60 and 40, whereas for 70S it is 50S and 30S. Now you must be wondering that what do you mean by S? So over here, what do you mean by S basically, beta? Can anyone decode for me that what do you mean by S? So S over here stands for Swedberg's constant. Swedberg constant after the name of the scientist, or you can also say basically beta sedimentation coefficient. So this S actually gives you the indirect measure of the density and the mass of the ribosomal unit. I already told you in the beginning that you don't have to add the two units. You don't have to make like 60 plus 40 means 100s. Yeah, 50 plus 30 means 80s. No, not like that size. Yeah, sorry. It's density and size, not, I'm so sorry, I used the word mass. Exactly, density and size. So it gives the indirect measure for the density and size of the ribosomal units. So you don't have to add. It's basically the density. It's the indirect measure of the densities of the ribosome. So you don't have to add and give the exact ki bhai 60 40 matlab 100 ho gaya, 50 30 matlab ho gaya 80. So you don't have to say that over here. Correct? I hope it's clear to everyone. So here S stands for sedimentation coefficient, which gives you the indirect measure of density and size. I hope it's clear to each and every one of you. The diagram is also clear. So when the two units are brought together, then it becomes the composite unit. This is called as ribosome. And again, this is used for protein synthesis. And that is why here also ribosomes are referred to as the protein factories. Clear? Now proceeding with cytoskeleton. Now remember, this topic is only exclusive to eukaryotes. Prokaryotes do not show cytoskeleton structures. Cytoskeleton are nothing but beta. Rather, these are just the minute, thin, proteinaceous, thread-like filamentous structures which are found scattered in the cytoplasm of eukaryote. इनका मेन काम होता है कि पूरे यूकैरियोटिक सेल का स्ट्रक्चर फ्रेमवर्क आउटर फ्रेमवर्क को मेंटेन करके रखे और एल्स दे कैन आल्सो हेल्प इन प्रोवाइडिंग मोटिलिटी टू द सेल और एल्स दे कैन आल्सो हेल्प इन प्रोवाइडिंग सेल डिवीजन. सो साइटोस्केलेटन इन यूकैरियोट can help in cell division process. It can help for motility purpose. It can also help in maintaining the overall structural framework of the eukaryotic cell, but not in prokaryotes. So what are cytoskeletons? Cytoskeletons are nothing. So cytoskeletons are nothing but the proteinaceous structures which are found scattered in the cytoplasm. Now these cytoskeleton can be of three types. So basically beta, cytoskeleton can be of three types: microtubules, microfilament, and intermediate filament. So they can be of how many types? Three types, right? Microtubule, microfilament, and intermediate filament. Microtubules, they are made up of which kind of protein? Tubulin protein. So tubulin proteins, they join together to form microtubules. Microfilaments, they can be made up of two different types of proteins like actin and myosin. Similarly, intermediate filaments, they are made up of lamin protein, the non-contractile lamin protein. Intermediate filaments, they're exclusively found in the inside the nucleus of the cell. Microfilaments, they are usually, by the name you can remember, actin and myosin. Actin and myosin, they are responsible for muscular contraction and relaxation. So they're actually responsible for muscular contraction. They also help in amoeba. They help in pseudopodia formation. Then during cytokinesis of animal cell, they help in that inward movement of the plasma membrane. So they help in the constriction of plasma membrane. During cytokinesis of animal cell, constriction of plasma membrane during cytokinesis of animal cell. ठीक है. अब अगर हम माइक्रोट्यूब्यूल की बात करें बेटा तो ये बहुत इंपॉर्टेंट है, इन पे ही हमें मेनली फोकस करना है, बिकॉज दीज माइक्रोट्यूब्यूल्स दे हेल्प इन फॉर्मिंग स्पिंडल फाइबर्स ड्यूरिंग द सेल डिवीजन. So they help in the formation of spindle fibers. Therefore they help in the movement of chromosome, right? Not only this, they also make centrioles. So they also help in cell division like this. Apart from this, they also prepare cilia and flagella. So that means they also help in locomotion, right? an intermediate filament inside the nucleus. They maintain the structure. Ma'am, what about WBCs since they are amoeboid shape? Amoeboid over there, Praneeth, means irregular shape. Here, pseudopodia formation is amoeba is a unicellular eukaryotic organism which is actually for its locomotion. It relaxes its plasma membrane and forms a false feet called pseudopodia. So that relaxation of the membrane, and once the work is done, so it again pulls back its plasma membrane, so that pulling inside and forming pseudopodia, this is the role of microfilament, right? However, WBCs, they are of that shape itself. They are not for forming pseudopodia. They are not showing that contraction relaxation continuously. They are of fixed shape, which is of amoeboid shape. So WBCs का shape ही fixed है, amoeboid, right? Irregular shape के होते हैं. However, amoeba उस irregular shape में रह के भी pseudopodia कभी इधर से निकाल देगा, कभी उधर से निकाल देगा, इधर से contract कर देगा, उधर से relax कर देगा. So this continuous contraction and relaxation to form the pseudopodia is done by microfilaments present on the membrane. Correct? I hope I am very much clear to each and every one of you. If yes, then now just remember two basic more points over here, beta. That if I talk about the structure of centrioles, centrioles are again the non-membrane bound cell organelle which are exclusively found only and only in animal cells. Their main function is to initiate cell division. In plant cells, they are absent. Now, they are usually, beta, two centrioles which lie perpendicular to each other like this. So, there are two cylindrical perpendicularly placed centrioles which are surrounded by an amorphous fluid called as centroplasm or kinoplasm, and there's no membrane around them. So single is centriole, and this whole two centrioles plus the fluid without any membrane is called as centrosome. They are made up of microtubules which are arranged in 9+0 manner. Correct? Now these centrioles, they themselves give rise to basal bodies. So centrioles are 9+0 microtubular arrangement which give rise to basal bodies that are also 9+0, and from the basal bodies arises the locomotory organ like cilia or flagella, but they are not showing 9+0, rather they show 9+2 microtubular organization. Now let's see what is this 9+2 organization in case of cilia and flagella. चलो तो इसको हम जल्दी से देख लेते हैं. तो बेटा cilia, flagella, unlike centriole, they are bounded by plasma membrane. तो ये रहा मेरा plasma membrane. Correct? Space inside the plasma membrane is called as axoneme. So plasma membrane के अंदर की जो space होती है उसे हम कहते हैं axoneme, where in arrangement of 9+2, where 9 means 9 doublets on the periphery. 2, 3, 4, 5, 6, 7, 8, 9. So 9 sets of doublet on the periphery, and center has one central doublet connected by central bridge and covered by central sheath. Now these all central doublets, they are connected to the center with the help of spokes. Okay, so there are 9 radial spokes. Apart from this, they are labeled as AB. इनको हम AB की तरह label करते हैं, and they are connected to each other also by a AB linker. ठीक है, like this. साथ ही साथ में जो B वाला हमारा है, on that it also have some structures which are called as arms. These arms are made up of dynein protein which provides energy for the locomotory part. So this is how the cilia and flagella beta basically looks like. Now the last topic which you have to see over here is now the nucleus. So what is nucleus? nucleus is nothing but the brain of the cell, which was first discovered by Robert Brown. किसने खोजा था इसको? Robert Brown ने खोजा था. This is also basically beta double membrane bound, but both the membranes are fused. Double membrane bound, which are fused, because of which there are certain openings. Okay. Now the number of nucleus in the cell can be highly variable. Some may have one nucleus per cell, some may have two nucleus per cell, like for example you see in case of paramecium, or there can be many nuclei also present. Correct? And there are some cells like mature RBCs and sieve tube which do not have nucleus as well. So if I talk about the structure of nucleus, which is double membrane bound, I told you both the membranes, they are fused like this, because of which there are pores present called as nuclear pores. ठीक है? तो ये हो गया हमारा outer membrane, ये हो गई हमारी inner membrane, which are fused, because of which you can see nuclear pores. These nuclear pores actually allow the inward and the outward movement of material through the cell and the through the cytoplasm of the cell and the nucleoplasm of the cell. Correct? Now the fluid which is present inside the nucleus, it is called as nucleoplasm. Always remember, the outer membrane is continuous with ER. Apart from this, the outer membrane may also have ribosomes attached. So these are the ATS type of ribosomes which are found attached to the outer membrane of the nucleus, but inner membrane is more selectively permeable and does not have any such attachments. It is smooth. Now this beta nucleoplasm, you can see it can be divided into three parts. The intermediate filament, which is present towards the peripheral part of the nucleoplasm, this is called as nuclear lamina. So this nucleoplasm can be divided into three parts. The peripheral part, जो बाहर की तरफ है, that is called as nuclear lamina. This is the part which is made up of intermediate filament. Correct? Then you have thin thread-like structures, colored structures called as chromatin fibers, which was first observed by scientist Fleming. These are actually the chromatin fibers which carries the genetic material in the form of DNA, RNA, histones and non-histone proteins. When cell division occurs, these thin thread-like structures, they get condensed to form thick rod-like chromosome, right? हाँ, बहुत बढ़िया चंदू, well done. Apart from this, they have a dense stained non-membrane bound structure called as nucleolus. This is non-membrane bound, densely stained structure called as nucleolus, whose main function is the active synthesis of rRNA, which goes out, joins with the protein and makes ribosomes later. So the number of nucleoli per nucleus is highly variable. It can be one, it can be many, depending upon the requirement of protein in the cell. अगर किसी सेल को ज़्यादा प्रोटीन चाहिए तो वो ज़्यादा न्यूक्लियोलाई रखेगा ताकि वो ज़्यादा से ज़्यादा rRNA बना सके, which goes out in the cytoplasm, joins with proteins and make the more number of ribosome. More ribosome, absolutely more protein synthesis. So this is how the nucleus beta appears to be. I hope things are absolutely, all the things are absolutely crystal clear. So with this, we come to the end of the chapter. So the last topic was your this nucleus, and I just hope everyone has enjoyed the lecture, and within very less duration of time, I have tried to give you each and every important concept of the chapter. We haven't left anything. I have tried to cover as much as possible in this span of 2 hours. And yes, if you go through this lecture for a quick revision, it is going to be very helpful to each and every one of you. So yes, thank you so much for watching. See you in the next lecture. Till then, bye bye everyone, take care.